Hello and welcome to Designs for Health Genomics Spotlight Educational Series. Today we'll be covering Part 1B of nutrigenomics. So if you haven't watched Genomics Spotlight Part 1A, please do so beforehand. And without further ado, let's begin. Multifactorial conditions can strongly influence the development of conditions or diseases. One excellent example is type 2 diabetes. This example shows a jar. The idea is people born with a very specific size jar have different risks of developing certain conditions. And there are environmental factors, which are the orange triangles, and there are genetic factors, which are the dark purple circles. So the more genetic factors you have, the less room you're going to have for environmental factors before you fill up the jar, at which point disease begins to manifest. And the same is true in reverse. The more environmental factors you have, then the less room there is for genetic factors, risk SNPs, before disease manifests. The bottom example in the left is a jar. And this shows unaffected persons with a certain size jar. And of course, the bigger the jar, the less chance you're going to have a certain disease. And as you can see, the genetic factors are the purple balls, and the further you go to the right, you can see the accumulation of orange triangles, and that is environmental conditions, factors, etc. And once the jar is full, that's when disease begins to manifest. Everyone has a different size jar for each disease or condition, and everyone has a different number of genetic balls in the jar to start at birth. So what you want is a bigger jar and or a fewer number of purple genetic balls, so to speak. Multifactorial inheritance is a key player that influences whether or not someone gets a condition or a disease. Some conditions, there's little or nothing you can do about acquiring them, but you can use nutrigenomics, microbiome, and metabolomics to manage these conditions or diseases. Here are the key characteristics of multifactorial inheritance. These characteristics include polygenic in nature. Multifactorial traits are determined by the combined effects of multiple genes, each contributing to the overall phenotype. Genes can have multiple alleles or SNPs that influence the trait, and the interaction among the different variants, called haplotypes, can lead to a wide range of phenotypic variations. Environmental influence. Environmental factors such as diet, lifestyle, supplements, exposure to different toxins, and other external influences can play a significant role in determining the expression of multifactorial traits. Complex traits and diseases. Multifactorial inheritance is commonly associated with complex traits and different types of conditions and diseases that simply don't follow simple patterns of inheritance. Examples include height, intelligence, susceptibility to common diseases like diabetes, cardiovascular disease, and certain types of cancer. And finally, population variability. Multifactorial traits often show continuous variation across a population rather than clear-cut categories. This variability can be influenced by both genetic diversity and the range of environmental exposures. The process of turning our genes into functional products like proteins is what is known in biology as the central dogma of biology. The outline of the process of how genetic information is transferred and expressed in living organisms is as follows. First, DNA carries the genetic information, as we all know, in the form of a unique sequence of nucleotides or letters, and it's located in the cell's nucleus and serves as the primary storage of all our genetic information. Transcription is the first step. So with transcription, DNA turns into RNA. And this is the central dogma first step. And during this process, the genetic information in a specific segment of DNA is transcribed into a complementary RNA sequence. The RNA molecule is called mRNA, or messenger RNA. RNA is a single-stranded molecule and acts as an intermediate between DNA and proteins, and it takes the information from the nucleus to outside the nucleus to the ribosomes, which is found in the cytoplasm, and the ribosomes make proteins. Translation. The final step is translation from mRNA information into proteins. During translation, the sequence of nucleotides in the mRNA strand is translated into a sequence of amino acids, the building blocks of proteins. This process, as we said, occurs on the ribosomes. In the context of genetics and genomics, DNA can be broadly categorized into two main types, protein coding and non-protein coding DNA. Additionally, within the non-protein coding DNA, there are different types of functional non-coding RNAs, such as microRNA and long non-coding RNA. For protein coding DNA, that DNA refers to regions of the genome that contain genes that encode for actual proteins. These genes are transcribed into messenger RNA, which is then translated into specific amino acid sequences, and eventually proteins are produced for various cellular functions. Interestingly, protein coding genes make up a relatively small proportion of the total human genome, estimated to be somewhere in the 1 to 2% range. However, these regions are critical for the synthesis of all proteins necessary for the proper functioning of our body. Non-protein coding regions of the DNA obviously don't code for proteins. However, they are also very important. There are a lot of functional elements involved. For example, promoter regions, enhancers, repetitive sequences, and non-coding RNA genes. Science hasn't quite yet grasped all of the intricacies of the human genome, including the reason and function for the existence of all these different types of DNA. But they're working hard to do so. As far as microRNA is concerned, it's a class of small non-coding RNA molecules. We're talking somewhere between 20 and 25 nucleotides long, and they're transcribed from specific genes called microRNA genes or miRNA genes. And when miRNAs bind to their complementary sequences on mRNA, they actually inhibit translation or induce degradation of the mRNA, leading to decreased expression. So in general, the more microRNA is produced, so if you upregulate some microRNA gene, it's going to inhibit some other gene. MicroRNAs are very important. They are important for immune response, cell cycle control, including oncology. They've been associated with various different types of diseases and neurological disorders. And finally, we'll touch upon LNC-RNA, long non-coding RNA. LNC-RNAs are a very diverse group of RNA molecules that are a little bit longer than microRNAs, and they also don't encode proteins. Like microRNAs, they play important roles in gene regulations and various cellular processes. LNC RNAs can interact with DNA, RNA, as well as proteins to modulate gene expression. They can also modulate chromatin structure and several other cellular functions. They've been implicated in numerous biological processes, including epigenetic regulation, which is a pretty big deal, transcriptional control, and cellular differentiation. Some LNC RNAs have also been associated with some diseases and may serve as potential biomarkers or therapeutic targets. In summary, DNA can be divided into protein coding or non-protein coding. Let's look at some of the different types of RNA molecules. miRNA, also called microRNA, are small RNA molecules that play an essential role in post-transcriptional gene regulation, a process that controls the expression of genes after they have been transcribed into messenger RNA. Both siRNA and miRNA are part of a broader class of small non-coding RNA molecules that don't actually code for proteins themselves, but they still regulate gene expression. siRNA, or small interfering RNA, are double-stranded RNA molecules, typically around 20 to 25 nucleotides or letters in length. And siRNA are involved in the process known as RNA interference, which is a natural mechanism cells use to silence or knock down the expression of specific genes. In RNAi, or RNA interference, siRNA binds to a protein complex called the RNA-induced silencing complex, RISC. One of the strands of the sRNA, known as the guide strand, directs RISC to target and bind to complementary sequences on the mRNA of the target gene. This binding leads to mRNA degradation or translational repression, effectively reducing the expression of the targeted gene, so it inhibits the gene. And here's another way of showing the DNA to RNA to protein pathway. In this second image, we can see that the introns in the original DNA and RNA don't quite make it into the final protein. Instead, just the exons do. You would think that between introns and exons, introns would be the segments that made it because they're in, and exons, maybe EX means out in Latin, so they don't make it. But it's actually the opposite. Exons is what comes out of the entire DNA chain. So exons are the ones that make it, and introns are the ones that are cut out. So you can see the 3 introns in this example don't quite make it into the final mRNA. It's just the exons. Indeed, the final protein is made up entirely of exons that have been spliced together. Our cells have a very effective method of turning the information in our genes into functional products, usually proteins or glycoproteins. And this constitutes the central dogma of biology: DNA, RNA, functional proteins. There are small but important differences between DNA and RNA. DNA serves as a genetic material and carries hereditary information. RNA has various other roles in our cells, including mRNA, which carries the genetic info from the DNA to ribosomes, transfer RNA, which carries amino acids to the ribosomes, and finally, ribosomal RNA, or rRNA, which is actually a major component of the ribosome itself and helps catalyze the peptide bond during protein synthesis. And if you look to the right, you'll see the images of DNA versus RNA. DNA is double-stranded and contains deoxyribose as the sugar, whereas RNA is usually single-stranded and contains just ribose as the sugar. And finally, in DNA, there are 4 sugars, thymine, cytosine, adenine, and guanine. With RNA, it's just like DNA, except the thymine is replaced with uracil, or the letter T is replaced with the letter U. DNA is relatively stable and has a long lifespan, allowing it to serve as a permanent store of genetic information. When forensic scientists find DNA, it usually is a lot more present than RNA because it lasts a lot longer. RNA is also very, very subject to degradation by cellular enzymes. And DNA is much more durable. Since DNA is a double-stranded molecule, it provides it with additional stability as opposed to single-stranded RNA. In addition, DNA has the base thymine, which RNA does not. Thymine has a methyl group, and that methyl group actually makes the DNA more stable and less susceptible to damage. And there are biological reasons too. DNA's role as the long-term storage of genetic information contributes to its longevity. Whereas RNA's primary function is to act as a temporary copy of the genetic information stored. Since RNA is not meant to last, cells do not invest in the same resource into repairing and preserving RNA as it does DNA. Genetic mutations can result in various types of structural changes in the DNA sequence, including deletions, duplications, inversions, and translocations. Now let's take a look at some of these different mutations. We'll start with deletion. Deletion removes a chromosomal segment. So as you can see here, D is the chromosomal segment that is disappeared or deleted after the deletion. Duplication is different. So here in the example, you see the segments A, B, C, D, whereas the example on the right, the segment BC is duplicated. So you have A, B, C, B, C, D, E, F, and so forth. During an inversion, this reverses the segment within a chromosome. So A, B, C, D, E becomes A, D, C, B, E. Sounds like kind of a tongue twister, but it just reverses the direction. A translocation is different. In a translocation, it moves a segment from one chromosome to another non-homologous one. So AB from this chromosome moves down, and now it's part of this other chromosome. And MNO from that second chromosome does a reciprocal translocation and moves up to the top where the AB was. In the context of genetic testing and variant classification, different terms are used to describe the potential effects of different genetic variants on a person's health. Pathogenic is one such term, a genetic change that is associated with a specific disease or condition. This directly causes or significantly increases the risk of developing the particular disease or health problem, typically associated with a well-defined clinical phenotype and have been shown to have a clear and significant impact on health. Likely pathogenic is not as strong, but it's still strongly suspected to cause the specific disease, but may require additional evidence or functional studies. Likely already has a significant amount of supporting evidence, but a little bit more additional data would be beneficial for a definitive classification. VUS, or variant of uncertain significance, is not so clear-cut. Clinical significance is not yet fully understood. It is definitely implicated, but quite a bit more data is needed. Likely benign is on the other end of the spectrum. It is not likely to have any significant impact on an individual's health. However, more research is still required to be confident as to its classification, so it's probably harmless. And the last one, benign. Benign variants have been adequately demonstrated to have no adverse effect on an individual's health. It should be harmless. Let's talk about histones. Histone modifications are chemical alterations made to histone proteins that help regulate gene expression and chromatin structure. Histones are proteins around which DNA is wrapped up, kind of like a yo-yo, and histones get together and form nucleosomes. These modifications do not alter the DNA sequence itself. Some common histone modifications include acetylation, which is just the addition of an acetyl group to specific lysine residues on histone tails, which is generally associated with gene activation, as it opens up the chromatin, making DNA more accessible. Methylation is another type of modification. It's the addition of, you guessed it, a methyl group to specific lysine and this time also arginine residues, which can either activate or repress a gene, depending on the specific residue and the number of methyl groups added. And finally, phosphorylation, the addition of a phosphate group to specific serine or threonine residues involved in various processes, including DNA repair and mitosis. As you can see in this pie chart, protein coding genes make up only 2% of the whole genome. This 2% is what traditional genetics is concerned with. However, modern-day genomics looks at not just the 2% protein coding genes, but everything in between. If you have a mutation in an intron that doesn't even make it to the final protein structure, that can still affect protein function and activity because the way proteins fold is affected by the presence of variants inside of introns. Now let's briefly look at a couple other parts of the genome. Lines are long interspersed nuclear elements. These are a type of retrotransposon that is found in large numbers in eukaryotic genomes, such as humans. Lines are transposable elements, which means they can move around the genome. They're capable of copying themselves via an RNA intermediate and then reinserting themselves back into the genome. Signs, on the other hand, are short interspersed nuclear elements. These are also a type of retrotransposon and are shorter than lines. Signs don't code for any proteins, but what they do is they hijack the machinery of lines to replicate and insert themselves back into the genomes. And for example, LTR retrotransposons are long terminal repeat retrotransposons. That's what it stands for. These are another type of transposable element, and they have direct repeats of DNA sequence at each end, the long terminal repeats. They're called retrotransposons because they replicate through a process that is similar to that of retroviruses. They transcribe their DNA into RNA, then use a self-encoded enzyme, reverse transcriptase, to convert the RNA back into the original DNA, which can then insert itself into a new location in the host's genome. A Manhattan plot is a scatter plot commonly used in genomics. It's named for the resemblance to the skyline of a big city such as Manhattan, with skyscrapers of different heights. In a Manhattan plot, the x-axis represents the genome divided into its constituent chromosomes. So maybe this would be chromosome 2, chromosome 10 to 15 would be right around here, and let's say this might be chromosome 20. The y-axis, on the other hand, represents the strength of the association between a genetic variant and the trait of interest. This strength of association is usually expressed as the negative log 10 of the p-value from a statistical test like a t-test. The reason for using this is the transformation allows very small p-values, which is very strong evidence of association, to be displayed as large positive numbers on the plot. This makes it easy to see which variants are the most strongly associated with the trait or disease. And over here on the right, you can see that we zoomed in on this green segment, and all the different dots represent SNPs, and the one with the highest significance, or the lowest log base 10 p-value, is the lead SNP. And everything below this dotted line is not significant to whatever level of significance the scientists decided to determine. And these SNPs, the highest one at the top is most likely involved in a change in the structure and function of the protein or enzyme. And there may be a haplotype, maybe 4 or 5 of these SNPs are associated with each other. And these ones down here, they might be guilty by association, since they may be in linkage disequilibrium with some of the SNPs here at the top, possibly even the lead SNP. Lead SNPs serve as markers to indicate a potentially important genomic region for further investigation. Once scientists discover a lead SNP, they can perform additional analyses to understand the functional implications of this lead variant and its potential role in the trait or disease. So to summarize, in this second section of Introduction to Nutrigenomics, we looked at genetic versus non-genetic factors. We analyzed multifactorial inheritance. We assessed the central dogma of biology. And we took a brief look at the structure and function of DNA, RNA, different types of RNA like microRNA and lncRNA. We looked at introns, exons, and splicing. And we know that exons end up in the final mRNA that turn into the proteins, and introns are cut out or spliced out. We discussed post-translational modifications to the protein. We looked at ribosomal RNA and tRNA or transfer RNA. We touched upon different types of mutations, including deletions, duplications, inversions, and translocations. We looked at the differences between pathogenic versus benign variants and everything in between, including classification. We analyzed histone modifications and their effect on the human genome and protein function. And we looked at nutrigenomic research and Manhattan plots. Thank you very much, and we look forward to seeing you in the next presentation.